Hello everyone. We are group 14 and our topic is monosodium glutamate. This will be our content. My name is Li Guangya and I'm gonna give a brief introduction to MSG. So first of all, what is MSG? MSG is a sodium salt of glutamic acid, a natural amino acid discovered in 1908 by Dr. Kikunai Ikeda from kelp. It is now produced sustainably via microbial fermentation. Its chemical formula represents a white crystalline powder. The discovery of MSG marked a significant milestone in the food industry, providing a means to enhance flavor in a variety of dishes. Recognized globally as a safe flavor enhancer, MSG delivers umami, the fifth basic taste, making it essential in modern food industries for boosting savory flavors without overpowering dishes. MSG's role in the food industry has expanded worldwide thanks to its ability to enhance umami without overpowering other flavors. Next, I'm gonna introduce the physical and chemical characteristics of MSG. MSG appears as an odorless white crystal or powder with extremely high water solubility, but is insoluble in ethanol. This characteristic makes its application in food very extensive. MSG remains stable at temperatures not exceeding 120 degree, making it an ideal choice for cooking and food processing, ensuring its properties remain unchanged during the production process. The low high-growth capacity of MSG reduces clumping, while its high purity ensures consistent performance during use, providing reliability for food processing. 1% MSG solution has a neutral pH value, which ensures its compatibility with various recipes and does not affect the acid-base balance of food. Next is MSG's functional benefits. MSG enhances umami 100 to 200 times stronger than salt, enabling 20% to 40% sodium reduction. With only 3 kcal per gram, it supports low-calorie diets. Produced via non-GMO fermentation, MSG meets clean label demands. Unlike artificial additives, it synergizes with natural ingredients to amplify meaty, savory notes. Cost-effective and efficient. 0.1 to 0.8% MSG optimizes flavor in snacks, soups, and processed meats, reducing reliance on salt, sugar, and fat while maintaining taste appeal. Finally is MSG's global safety recognition. MSG is globally recognized as safe by FDA, GRAS, EFSA, GECFA, and so on, with no daily intake limits, means linking it to Chinese restaurant syndrome. It is lack scientific validation. Rigorous studies confirm its safety for general use.
Hello everyone, I'm Xiong Yuanqi. I'm gonna introduce the cell physiology and product synthesis of MSG. The first part is the physiological role. Glutamate is the primary excitatory neurotransmitter in the central nervous system, activating postsynaptic receptors. The second one is the metabolic intermediate, participates in the TCA cycle and serves as a fixer for other amino acids. Now let's talk about the mechanism of action. MSG binds to T1R1, T1R3 heterodimeric receptors to taste buds, triggering calcium-ion dependent signaling cascades via G protein activation. As a transporter, glutamate enters cells via excitatory amino acid transporters driven by these ATP gradients. As the metabolic fate, It converts to alpha ketoglutarate or glutamine. Excess glutamate is cleared by astrocytes to prevent excitotoxicity. The second part is the industrial production of MSG. MSG is produced via aerobic fermentation using Corynebacterium glutamicum. The first step is stream selection and culture. High-yield mutants are used to enhance glutamate secretion. The second step is fermentation. For carbon source, glucose or molasses finally turns into TCA cycle intermediates. For glutamate synthesis, GDH converts alpha-ketoglutarate finally goes into L-glutamate. Biotin limitation increases membrane permeability, promoting glutamate effects. The third part is downstream processing. Ion exchange chromatography isolates glutamate from fermentation broth. For neutralization, glutamate aced with sodium hydroxide finally turns into MSG, and finally goes to crystallization and driving. Pure MSG crystals are obtained. Now let's talk about the metabolic engineering advances. The first one is NADPH optimization. Overexpression of PPP is a means to supply reducing equivalents. The second one is fermentation control. We control pH for 7.0, temperature for 30 to 37 Celsius, and erections are tightly regulated.
Hello everyone, my name is Lu Jinghua. Let's move to the third part, upstream fermentation processes of MSG. I will divide it into three parts, strain selection, medium optimization, and fermentation control. The first one, strain selection and physiology. The primary industrial strain for MSG production is Corynebacterium glutamicum, genetically optimized for high glutamate yield and good thermal tolerance and pH resistance, and reduced byproduct formation via metabolic engineering. The next part is medium composition. The fermentation medium must balance cell growth and glutamate synthesis. The carbon source is sugarcane molasses and cornstarch hydrolysate. The nitrogen source is urea ammonia, and it must be fat patch to avoid inhibition. The minerals can be magnesium ion, ferrous ion, and potassium ion. They act as the enzyme cofactors. And there is another, biotin, acting as the growth factors. The next part, fermentation process control. We can see from this flowchart, it tracks the biological timeline. From inoculation to harvest, we manipulate nutrients and pH at each metabolic phase. In the batch phase, it is about the cell's growth. We have to make sure the dissolved oxygen maintained at 30%. The fat batch phase is about dynamic glucose urea feeding. The residual sugar must be below 1%. Production phase, the pH at this time shifted from 7 to 6.5 to induce glutamate secretion. Temperature is kept at 32 to 34 degrees to minimize byproducts. And we have some critical parameters and optimization methods. To achieve cost-effective MSG production, we focus on three metrics. Duration. Between 24 and 36 hours balances yield and sterility. Yield. 80 to 120 grams per liter glutamate. It turns out to be over 60% conversion rate. The efficiency part, we put real-time DO and pH sensors for automated feeding. The final slide, we focus on the industrial equipment chain. We can see from this real picture, we can recognize the factory workflow, how lab-grown cells move through seed tanks and to the industrial fermenters, ensuring seamless scale-up. And we finally move to the brew's output, and they are sent to downstream.
Today, I will be presenting part 4, focusing on the downstream separation processes for monosodium glutamate. The downstream separation process includes all the critical steps following fermentation. Its goal is to efficiently extract, purify, and obtain the final MSG product with high quality and purity. After fermentation, the broth contains microbial cells, unreacted substrates, and various byproducts. To begin purification, we use centrifugation or microfiltration to remove biomass and large particles. This step ensures that only the soluble components, including glutamate, move forward in the process. Next, we address color impurities. Fermentation broths often contain unwanted pigments. We treat the broth with activated carbon, which effectively absorbs these colored components and other organic contaminants. Once the broth is clear, we purify MSG using cation exchange to remove unwanted ions and electrodialysis to improve purity. Then we crystallize MSG by adjusting the pH to 6.2 to 6.5 and cooling the solution to 10 to 10 degrees Celsius, which promotes crystal formation. The formed crystals are separated from the liquid using centrifugation or filtration. Next, we wash the crystals to remove remaining impurities and then dry them, either with hot air or under vacuum conditions. Finally, we dried MSG is sieved for uniform particle size and packaged under hygienic conditions for distribution. Throughout the process, certain parameters plays a critical role. Temperature affects solubility and crystal size. Lower temperatures improve crystal formation. pH must be tightly controlled to maintain MSG in its proper form. Incorrect pH lowers yield and purity. Ionic strength influences solubility. Optimal levels help enhance crystallization while minimizing impurities.
My name is Ding Kaiyuan, and in this part, I will talk about the quality control. I think this step is not at the end of the process because if by doing so, it will be too late for the quality control because the content might go banned at any step of the whole process. So the concept of quality control is actually starts from the beginning and to the end when the process is done. In this part, I will provide some key parameters or messages for the MSG quality control. So first we can see the raw materials. Normally cornstarch or molasses are used to provide the efficient nutrition for the culture to grow, and this has to be abundant and also safe and clean enough. So you have to check about the purity to make sure it can provide enough nutrition. And also, for example, you have to check no metal ion, no microorganism which may produce the acid. The acid will ruin the medium by changing the pH to a very acid environment. Then the nitrogen source standards is about the liquid ammonia and also urine. They have to be checked about their purity as well, because those are the only two provider in the whole process to give nitrogen for the cell synthesis to actually make MSG. So and also the sterilization. This is definitely needed because otherwise the nutrition will be consumed. As I said, if there's some bacteria, they will consume the nutrition and also generate the acid. Okay. Then the next part, the strengths. The strengths are needed to be checked about the homology. This step is to guarantee the enzyme, like the enzyme for the hydrogen or any other enzymes to guaranteeing them they won't lose their activity during the process. So there are you can see the parameters, the temperature, and also the message using to confirm the strength purity. Okay, and in the fermentation control, you have to make sure the concentration of the dissolved oxygen, also called the DO, to be very precise and in each step of the process. Otherwise, maybe the cell is broken. So, and also pH is important as well in this process. So, and finally come to the extraction and purification. During this step, this must be safe to make sure the quantity can meet the standards when those contents are made into production. Okay, I think that's all for this part.